Now that we've completed the series of videos that go into more detail about how we go from a gene on DNA to the protein that's encoded by that gene or coded for by that gene, we're going to move along and talk a little bit about genetic regulation of protein synthesis. Now, I do want to point out that your textbook covers this in a lot more detail than I am on this video lecture clip, and your textbook also includes more information than I require for testing purposes. However, you will be exposed to that information during your reading, so you've had some exposure to those topics, even though I'm not requiring the information for testing purposes. All right, so what do we mean by regulation of protein synthesis? And genetic regulation, what this is telling us here is, all right, so proteins are responsible for traits, but sometimes a cell needs to make those proteins and sometimes it doesn't. And that is regulated at the genetic level, because you've just seen how you go through these multi-step processes of transcription and translation if you need to make a protein. That requires a lot of effort by the cell. So if you don't really need that protein at a particular time, then it's better to shut down transcription if you can. It doesn't always work that way, but that would be the best way and the most conservative of energy. So we're going to talk a little bit about genetic regulation here. And again, your textbook goes into more details. It's going to explain an example type of genetic regulation in bacteria, which is good for you to read about, but I just don't require that information for introductory microbiology students who are planning careers in the health sciences area. All right, so this is a depiction of the chromosome from Escherichia coli, good old E. coli that lives down in your colon. So again, the chromosome is, a bacterial chromosome is generally a circular piece of DNA. It's going to be large, probably in the millions of base pairs in terms of the length. And it's going to have numerous genes on it that code for proteins that bacterial organism needs to use to survive. And so what they've done here, this whole circle is supposed to be the bacterial chromosome, and these little colored areas with these abbreviations, those are example genes that are located on that chromosome, and each of those genes has an abbreviation for its name. You know, for example, His here, that little spot right there, that contains the DNA that probably codes for an enzyme that's responsible for making the amino acid histidine in bacteria. So bacteria are going to be able to make, you know, many of their amino acids, if not all of them. Lac right here, that little yellow area, that's indicating a stretch of DNA that contains a gene or genes on it that are involved in the use of lactose as a nutrient. So we've talked about that before. Some bacteria can ferment lactose and some cannot. So if you are going to be able to use lactose as a nutrient, you better have the genes on your DNA that code for the proteins that allow that to happen. All right, so on a typical bacterial chromosome, you would expect about 75% of the genes are expressed at all times. That means that those genes are coding for proteins that are so critical for survival of that bacterial cell that those proteins have to be made all the time. Proteins wear out. So they get manufactured, they're used, they do their jobs, but they wear out and they have to be broken down and digested and replaced. And so if you need particular proteins all the time, you're going to have to keep manufacturing those so that you have nice, fresh versions of them to do their jobs that they're supposed to do. Now the other 25% of the genes on a bacterial chromosome, those will be coding for proteins that are only need to be made on occasion or on an as-needed basis. And a good example of that would be these genes right here that we were talking about that are involved in using lactose as a nutrient. So if you don't have any lactose in your surroundings and you're an E. coli cell, why waste the energy and effort to make the enzymes that are going to help you use that lactose? So what you want to do is only express those particular genes if lactose is available as a nutrient. And that's something that's covered in your textbook in more detail. When you're reading about that, you'll see, learn a little bit more about how that works. And again, when I say genes are being expressed, I mentioned that in an earlier video clip. Gene expression means the trait associated with that gene appears. So you're going from DNA to RNA to protein to trait or phenotype. Now, lots of genes need to be expressed all the time. Now, if you think about it, again, not all genes on DNA code for proteins. Usually when we are thinking about this story, we're going from DNA to messenger RNA to protein to trait. But you've got those other kinds of RNA as well, like the tRNAs that are involved in translation, the ribosomal RNAs that are present in ribosomes, and you have these regulatory RNAs that have been recently discovered, and those are produced by transcription as well. So there are going to be genes for those RNAs on your DNA, and they're produced by transcription. Transcription makes RNA molecules. It's just that those other kinds of RNA don't go off and get translated on a ribosome. They have other jobs that they need to do. They don't specify proteins. But those types of things are you have to constantly crank those out. So those are going to be genes that are always expressed as well. Now, gene expression, gene regulation tends to become more complicated in eukaryotes, and especially when you get into a multicellular organism like a human being, where you've got all these different organs and organ systems that have specialized jobs. So this gene regulation story is much, much, much more complex when you get into a human being or in other types of multicellular complex organisms. Because if you think about it, you know, each of your cells in your body, the trillions of cells that you have, they all have the same chromosomes. So they've got the same genes located on those 46 chromosomes that are present in your cells. But think about how different the traits are among the different cell types in your body. You know, think about a skin cell in your epidermis. Think about a cardiac muscle cell in your heart. Those cells in your body, they all have the same DNA. They have the same sets of genetic recipes. But what makes a skin cell, or a cardiac muscle fiber, a cardiac muscle fiber, or a neuron in your brain, a neuron, is that, yes, all of those cells have some genes that are critical for survival and basic functions like cellular respiration. Those genes are always going to be expressed. But there are genes that are specific for a cardiac muscle fiber, or specific for a keratinocyte in your epidermis, or specific for neurons that are only going to be expressed in those particular cell types. In other cell types in your body, those genes are not expressed. They have to be kept under control and blocked from their proteins being produced. Okay, some other examples of gene regulation. You know, why are some genes expressed and others are not? And I've talked about enzymes for catabolism of particular nutrients. Ideally, you only want to make these protein enzymes here if the nutrients are available, and there are examples of how that works in bacteria and other organisms. Another interesting thing, pathogenic bacteria, so bacteria that can cause disease. You may remember back in chapter 3, when I was talking about the bacterial cell parts, I mentioned that, you know, fimbriae, the little spikes on the surface of bacteria, in some cases those are present on the surface of a pathogenic bacterial cell early in infection when those bacteria need to adhere or attach to a surface. But later during infection, after the bacteria build up their numbers, they stop expressing those fimbriae. Those fimbriae are built from proteins, so they're going to be genes on the DNA that code for them. You stop making new fimbriae, and the reason for that is because once the numbers have built up, it's more beneficial to the pathogenic bacteria to quit making those fimbriae because they're sticking out from the surface, and those are things that the cells of your immune system, your white blood cells, can use to help identify and attack those bacteria. So you'll see as we move later into the course that these pathogenic organisms are very clever and have evolved a lot of different interesting strategies for combating our immune system. It's a real battle that goes on between us and them. Same thing with capsules. There are some pathogenic bacteria that express their capsules early during infection, but then quit producing those capsules later if it's advantageous to them to try to help them avoid the immune system. Quorum sensing in bacteria, we talked a little bit about that. That's a great example of genetic regulation. We talked about how bacteria in biofilms will completely change some of their characteristics and their traits if they realize that they need to assemble a biofilm and participate in a biofilm community. They've got to turn on certain genes that help them make those sticky substances and help create the biofilm. So all that quorum sensing we talked a little bit about when we went over bacterial reproduction is an example of genetic regulation as well. So again, regulating protein synthesis at the genetic level, that is generally done through stopping transcription or stopping translation. Now you might think, well, why don't you just stop transcription? Because that's going to conserve the most energy. Why do transcription and make the messenger RNA if you're not going to go on and do translation and make the protein? And the best answer I have for that is that's just the way it works. Now in some cases, genes that don't get expressed, that is blocked at the transcription level, they just don't get transcribed. But in other cases, the gene gets transcribed, you have the messenger RNA copy of the gene, but the messenger RNA copy does not go on to be translated. And again, section 8.3 in your textbook, you will be reading more about how genetic regulation works. But for this course, I feel like that's, we're time constrained in a one semester microbiology course, and some of those concepts can get a little bit deep, and we're just not going to be able to completely cover that topic in this class. All right, so I'm going to conclude that video here. On the next one, we're going to move on to something that's different. We're going to talk about DNA recombination events and what that means, and we're mainly going to be focusing in on bacteria and some methods by which some species of bacteria can exchange DNA with each other. So that'll be the topic for the 9th video lecture for Chapter 8.